if you're wondering why I'm laughing, it has crashed twice for the real recap, APsolute recap. So I'm going to host it now and see what we can go from there. Sorry, guys. We really want to help you, we promise. And she has probably saved it on her side. So all the stuff we've talked about before was good to go. So, hi.
But hey, they keep coming back. We must be doing something right.
They love us.
Yeah. You have now. Good luck. You're on the questions.
But nothing down here, right? Oh my God, there's a ton of questions down here.
So some of them are going to be from your own feed from the last time you put a question box. It saves those. And some of them will come from the ones probably transfer over because we switched hosts. But
okay. So I see just now on there, which means I can pop that up.
It's going to cover my face. Can't wait. Guys, thanks for being super resilient and coming on back every time it crashes. We'll keep hosting as long as you have questions.
Okay. And also every time I can get the questions, if we don't get to them all, then I'll try to post them on my story so we'll at least have them later. So this question says, if you predict wrong but justify correctly, is it a loss of 2 points or can you still get 1? In the past, they have been separate points. But recently they started kind of merging those two and kind of marrying them together. And so you have to predict to justify. This is a brand new redesign and brand new exam. So there's no way for me to actually know how they're going to do it this year. I would, give your best foot forward, predict, and then justify that prediction. Don't contradict yourself. And then you should be good. Anything you want to add?
No, I mean, that makes the most sense. The points are, like we said, the points are adding up to make sure that they don't want to think you're guessing. I mean, at the end of the day, you got to prove that you know information. Did you guys go over when I was silent, the answer all the questions to get a 5?
We talked about that, and we mentioned that you could still get a 5 even if you don't have all the parts answered. And so this kind of couples and backs up to that one. They said, would it be better to attempt all the parts or thoroughly answer them? It's a points game. If you don't give enough detail, you might not be able to get your credit. So it would be best to give them what you got. And you have 25 minutes. You have more time than you traditionally would have on an exam. Anything you want to add?
Yeah. No, I mean, when we have, it's better to be specific and fewer questions, that's all you physically can do, than vague on multiples and not get the points at all. So definitely dive into the questions that you have and you're most confident in first from that point. I had a question earlier. I know you're going to pull up another one in a second, but it was like, what these last 48 hours? I'm so nervous. What can I cram? And you got to give yourself a little bit of chill time. Please, please, please don't sacrifice sleep for studying. This is not the time to do that. You've been studying all year long. If you're going to do anything these next 40 hours, do a couple timed FRQs. Put yourself in a time scenario so that you can practice more specifically and not kind of just meander about so much content.
Hey, I'm going to jump over to a content question, and then we can come back to more questions about the actual how to take exam. Because I really want to make sure that we're helping the kids. So what organelles do we need to know?
Which organelles don't you need to know? Let's ask it that way.
You probably need to know all of them. Yeah. You need to know the nucleus. And you need to have a general idea of what happens in each of these places. So, nucleus is where we're going to host the DNA, the difference between the rough ER and the smooth ER, and also understanding about how that leads to other things. So we know that the smooth ER is in charge of detoxification. So if they gave you a question in which they showed one cell has a small amount of smooth ER and another cell has a large amount of smooth ER, identify the function of this cell. You would then be able to say that the cell that has more smooth ER must be part of a detoxification process, and you could then build from there.
Oh, Hector.
Okay. Can I please hurt Hector?
Yeah,
Hector.
Don't take it personally. Don't take it personally.
That's my student.
Button.
That's my student.
Oh, is it really?
Okay. Yes. Also kind of thinking about,
for those who are not your student, what would you say to that statement?
that is a middle school statement. That does not hit AP level. I'm sorry, you will not get credit for that one. Hey, Betty's here. Sorry, I'm excited my students are here.
That's awesome, guys. And so part 3 of Q&A. I'm Melanie King from the APsolute Recap. Obviously, some of you know your own teacher who's here as well.
I'm Tiffany Jones. I think I'm a penguin. Okay, so here's another content question.
Let us know.
And I'm going to let you do this because you did the Operon the other day. Is she talking?
I don't even. All right.
There you are.
It might have.
Yeah.
All right. Prokaryotic.
You got it as your question of the day 2 days ago?
I did. That's true. All right. So expression is simpler, we'll say, than eukaryotic. And there's a couple reasons for that, because prokaryotes have a singular circular chromosome, whereas eukaryotes are going to have multiple levels of gene regulation, ranging from histones and methylation in the nucleus to transcription factors, onto mRNA processing, alternative splicing, and so on. But in the prokaryotic land, you have operons, and operons are sequences of genes that have regulatory regions. And essentially what's happening here is like an on-off switch. Either you're going to have RNA polymerase more likely to bind, which increases transcription and translation, or less likely. And that's a picture of all of these. It's really common too for the product of that gene expression to turn off the transcription as well. Okay.
You good? Okay, here's your new question. We know that Unit 6 is going to be on the exam, which is all that biotech stuff. What part of that is going to be most important for us?
Really, I think an understanding of how that kind of works. You don't have to know specific steps, but I can see them give you the results of gel electrophoresis or PR within a larger prompt. What do you think?
Or having the transformation in there and connecting that to some other component. Because you could then take about, some type of DNA that you've inserted into a gene, into the DNA of the prokaryote, and then you've done this, and this. Also, I'm going to kind of take a moment, and somebody mentioned about southern blotting on your question thing earlier, so I figure this is a good time we can kind of slide that in. Southern blotting, there are multiple different types of blots. You've got northern blot, southern blot, and then western blot. There's no such thing as an eastern blot. Northern and southern have to do with the nucleotides. So northern blot is your DNA. No, I'm backwards. Correct me. Southern blot is your DNA.
Yeah.
And northern blot is your RNA. And then the western is your proteins. And as you see, it's not even in the curriculum. And so I'm actually shaky on it. I'm sorry, guys. But they could give it to you and ask you to apply something from that. No, your math doesn't need to be in sentence form. If you're showing math, you could just show the math that you're doing. Sorry, I saw that question. A blot. I lost her. Where'd she go? Okay. So a blot has to do with, the best way I can think about this is, girls, when we put on our lipstick, we'll blot. We'll put like a piece of tissue in there, and then we will actually like blot and kind of get the residue off. And so after you run a gel, what you'll do is you'll put a kind of a piece of paper. It's a nitro cellulose paper on top of it. And when you put that on top of it, you can then pull components from the gel onto your paper, and so you've blotted it. And so it'll allow you to find specific bands that you're looking for. And so that's what we're talking about when we do a northern blot or southern blot or western blot. We're pulling those specific bands off. So You probably don't need to know specific methods like Sanger. I wouldn't be too worried about that. I also, real quick, you don't really need to know the difference between DNA polymerase 1 and 3. Just know that DNA polymerase is going to allow you to make that DNA polymer. Really important thing about DNA polymerase, you have to know the directionality that it reads and the directionality that it forms. And so in terms of DNA polymerase, it's going to read 3', which is where your hydroxyl is, to 5' where your phosphate is, okay? And then it's going to synthesize that DNA 5' where the phosphate is to 3' where the hydroxyl end is. And so it is very important that we know the directionality of DNA. But in terms of DNA polymerase, you don't actually have to know the difference between 1 and 3. That's past the curriculum. Also, you have to know that it can't build from nothing. So you need to know that there has to be some type of primer, or there's another DNA strand that's building off of. And so with like the lagging strand, there's going to be a previous strand that you can kind of, I'm sorry, with the leading strand, there's a previous strand that you're building off of. So there's that. So let's see if we can jump back to some questions about the actual. What's wrong?
Can you
okay.
hear me? Am I actually here?
You're back and forth. So the question is, can you annotate on the computer? You cannot, but you are allowed to print the question. So as soon as the question pops up, you're able to print it, and then you can highlight, annotate, underline, do whatever you need to do. But the actual software that the test is made with, you can't actually do anything. Is there anything you want to add, sweetheart? Can you
No,
talk?
I can hear.
Yes,
I keep. Oh, sorry. Can you hear me right now?
I can.
Okay. Last I was here was something with blotting. I was gonna talk about lipstick, and then I left. So now I'm back with DNA polymerase, I think.
Yes, I already talked about lipstick, so we're good. We think the same.
Great.
Okay, let me pull the questions back up again.
Sure.
So I guess we can do this one because like this is going to directly connect to the labs, like because they could probably pull one of the labs we've maybe done.
Mm-hmm. Yeah. So what labs apply to this year's AP exams? So it has to be something from unit 1 to 6, but you know they're recommended labs in the CED. In terms of required, what does the CED actually require from a lab perspective for teachers?
We are no longer required to do any of the labs. We just have to make sure that 25% of the course is lab-based.
Is lab. Yeah.
So, and actually the lab that they'll probably give you is something that you never did in your classroom. It's going to be some type of application that you could have done. Like it's an application of content and less about, hey, this is actually, hey, we took some potatoes and we stuck them in different colored solutions and we figured out what the concentration of the potato was. That's something you did in your class, but they're not going to give you a question like that. They may talk about grapes, and they may talk about putting grapes into, you know, salt water and say that it can cause an individual to have better wine or something. So they kind of will take something we've done and expand it somehow.
Yeah. And then next year, all teachers will be buying grapes and salt water and putting them in salt water.
They will.
I know. Animal behavior, no. Units 7 and 8. But here's the deal. If the College Board gives you like the definition of something related to ecology and then asks you a question following up on that, then that's okay. I mean, they can technically ask you anything. They just have to either tell you the information that you had to know in advance or provide you the information in the question that's specific enough so that you can reference back to it.
Sorry, I didn't want to lose the questions that I was finding. So somebody asked about cell signaling and communication. In terms of this, you're probably going to be given a question similar to, I believe it was last year's exam, in which they talked about interleukin and the gastrodermin. And so that one was an example of the cell signaling in which you were seeing kind of the pathway. So they're not going to just give you a generic receptor transduction response. They're going to give you a specific example, like the way that a cell is able to fight off an infection, and the pathway that takes place there. Anything you want to add?
Yeah, and what we talked about a little. Yeah, we talked a little today. They're not going to want to give you anything that's going to set you. So a lot of people have thrown out guesses that the coronavirus is going to be on the AP exam. Like, there's no way that's going to happen. So please don't put your brain space. Mechanism and steps of fertilization. So when it comes to reproductive cycles, you are not required to know specific reproductive cycles of plants and animals. But again, it doesn't mean that it can't be part of a prompt or question, and you have to apply your knowledge of meiosis to those questions. We mentioned this the other day, but remember that both chloroplasts and mitochondria are specifically inherited through eggs, and so they can show unique patterns of inheritance because of properties.
So going off of genetics and kind of spiraling there, they do want us to do a genetics problem, so we can maybe do that in a couple minutes. But there is a question about the checkpoints in mitosis, and this is kind of an important thing too, knowing the function of the checkpoints and then what happens if you bypass those checkpoints. Do you want to take this one, or do you want me to do it?
No, you're good.
Okay. So there's three main checkpoints that we have in mitosis. The first one is the G0. This has to do with whether you get the growth factor, whether you get the signal to cause the cell to enter interphase and start to divide. And so the second one we have is G2. This is going to determine that we have replicated our DNA, and that our DNA is, that there's no errors or no major errors in the DNA. And then the last one is where we get non-disjunction, which is the M checkpoint. It'll happen during metaphase, and if the microtubules are not attached to the kinetochores, and you bypass this M checkpoint, then it will cause non-disjunction, which ends up giving you extra chromosomes in a cell. So it is important that we understand about the checkpoints. Like, so cancer is able to bypass a lot of these checkpoints. And that's why we see a lot of the components of cancer. How does cyclin play a role? Cyclin allows you to get past the G2 checkpoint. Cyclin is going to slowly increase in concentration. And as it increases in concentration, it's finally going to hit a point where the cyclin-dependent kinase that binds to it then will allow you to go into the next phase, which is getting into mitosis. And so that's when we kind of see that step. Grading's all over relatives. She left me, so I have no one fielding my stuff. I don't think you need to know about alternation of generations. I don't really think they're also going to grade any easier. Like, we're going to grade the same way that we always grade. You need to have the content in there. I also don't think we're going to grade harder. I believe that the exam is going to be scored the same way that it's always been scored. The only difference is that we're going to be scoring them from home instead of at Kansas City where we normally grade them. You don't need to know about the immune system. Immune system is actually taken completely out of the curriculum. So if they give you a nervous system, endocrine, or immune system question, it means that it has to be, they have to give you background on it. And so they have to tell you the function of the immune system. They have to tell you certain cells that are in the immune system and how those apply. So they can't just ask you a straight, how does the immune system work? Also, they're probably not going to ask you that question since you have open notes.
I moved closer to my router. We'll see.
That's okay. So let's see what other questions we have going on. What is semi-conservative? So semi-conservative has to do with replication. And so in replication, we have two strands of DNA. They're anti-parallel. The two strands of DNA will come apart. And so you'll use each strand as a template. And so your daughter strands are going to conserve the parent because they have one parent strand and one newly formed daughter strand. Anything you want to add?
Yeah, so that's actually really important for PCR. When you think about the way that PCR works, the reason why the replication is so exponential, it has to do with the fact that DNA replicates in a semi-conservative way. All right. No, you do not have to know like CAM plants. That was part of the old curriculum, but not currently.
Pyruvate oxidation is having to deal with that when we oxidize pyruvate. And as a reminder, oxidation has to do with when something loses electrons. And so you see pyruvate in the mitochondria because, well, it's as it's moving into the mitochondria. And so that's part of cellular respiration. So you won't really see that in your chloroplast. I think she's frozen again. Okay. Do you need to know specific biotech? I would probably make sure that you could apply biotech. So as we said earlier, be able to apply the gel electrophoresis, be able to apply what PCR really is. And I think she was mentioning PCR a moment ago. With PCR, you're able to make multiple copies of DNA in a very short amount of time because you have a primer and you're also only using a very short sequence of DNA. It's also kind of, you can think about enzyme functionality when you think about PCR because the enzymes that you use in PCR are going to be heat resistant. So they actually came from thermophiles, which are bacteria that live in heat areas or very hot areas. And so because with PCR, you're going to heat it up, cool it down, you need to be able to have something that can not denature when it heats up. So let me see if I can see any other questions we have. Are there certain sections that are probably more important? I can't predict what's going to be on your exam. I can't tell you what's going to be more important, what's less important. I can tell you it's going to be unit 1 through 6. That is what we know, and I've got no insider information. I wish I did because I would love to share it with you, but I have no insider information for you. And we've mentioned this before, typing in versus paper pencil, it's all depending on what is best for you. I would practice with the online software and figure out what is the easiest way for you to be able to submit your answers. The questions don't come in PDF. They're literally just going to be, I believe, on your screen. But I honestly don't know. The demo didn't have a question in it, so it's hard for me to be able to tell you whether it was a PDF or not. These past few weeks, make sure you study tonight and tomorrow because your test is at 2 o'clock on Monday. How is a gradient formed and how does it lead to ATP production? So whenever we're talking about the electron transport chain is dealing with that we have high energy electrons. So we have these electrons that are high in energy, and as they fall down in energy, they're going to release some of that energy, okay? And that energy released is going to fuel a proton to be pumped across the membrane. And so that proton is going against this concentration gradient, so it requires that energy, okay? And so at the endoelectron transport chain, you're going to find there's this high concentration of protons on one side of the membrane versus the other. If we're talking about the mitochondria and cellular respiration, that high area of protons is going to be the inner membrane space. And then if we're talking about the chloroplast and photosynthesis, that high proton concentration is going to be in the thylakoid space. And so what ends up happening is that as it, whoa, she came back and I'm trying to get her back in. So as the protons fall back down the concentration gradient, they're going to release potential energy. And that potential energy is then used to turn a turbine, if you may, of the ATP synthase to synthesize ATP. And so actually NADH and FADH2 kind of are different energy levels. NADH has higher energy and FADH2 has lower energy. So you can make, I believe, approximately 3 ATPs per NADH and about 2 ATPs per FADH2. Let me see what other questions we got going on.
So I see a question from Damon in the chat. How much percentage do the tests need to solve to get a 5? That's actually not predetermined. So when the results come in, many tests are reviewed to finalize the scoring guide, and then the scores are distributed from that point forward. So it's the different amount every year, depending upon how the student responses are.
Nikki, I was wanting to know about if they're the same percentages. So when you look at the curriculum, which you guys can actually go and find, or the teacher handbook of the curriculum, just search AP Biology CED, and you'll see the percentages that originally were supposed to be on the exam, and they were never equal to begin with. So I don't know if the exam is going to be equal, really. I would
There's
probably
no points to play with at this point.
Yeah, and it's hard for them to, like, they can branch a lot of units between the questions, but there's no way for us to predict that. And even if I couldn't tell you.
Yeah.
Sorry.
I mean, unit 1 is so integrative into every other unit anyways. I mean, the chemistry of life. So that is probably going to be represented through other ways. So unit 1 likely won't be distinctly on there, but.
We talked about this Thursday when we did our pedigree stuff, and I'll post those pedigrees on my story later. So that anybody who needs it again, I can do that. But real quick, kind of thinking about the difference between dominant and recessive on a pedigree. A dominant allele is going to show up in every single generation. It cannot skip generations because it's dominant. Versus the recessive trait is going to be able to skip generations. You'll have an affected grandparent, an unaffected parent, and then an affected offspring. And then when you think about sex-linked versus autosomal, you're kind of looking at males versus females. A male only needs to have one affected X versus a female needs to have 2 affected X's. So like if you have an affected female and you see that the male is affected, there's a high chance that that's probably a sex-linked trait.
Yeah, remember autosomal is on chromosomes 1 to 22 for humans. Sex-linked being on chromosome 23 for those traits.
So that answers the pedigree question. Secondary messengers. So there's 2 main secondary messengers. You've got cyclic AMP and you've got calcium 2+. These are both the main kind of secondary messengers that we have. Big thing to know is that they're able to move throughout the cytosol to cause another component to the transduction pathway. So for example, if you think about muscle contraction, the calcium that's released from the smooth ER or the sarcoplasmic reticulum binds to a enzyme or a protein that changes shape, which then allows the muscle to contract. So it's like acting as another messenger in there. Again, they have to give you background information. They can't just assume that you know what's going to happen in this pathway. They have to tell you the calcium will bind to this thing, which will then do this. What would happen if there was a mutation that inhibited the calcium from being released? Or what would happen if the individual died and all the calcium was released? Like, they would have to give you background and then ask you a question about that. So it's kind of application of content, not just straight knowledge questions. Don't memorize. Yeah, don't memorize anything. Are the questions on the exam going to be long? I think that your long FRQ is going to be that long because number one, they're trying to push you. They want to get you to, basically they're pushing your time. They want to stop you from using the internet. They want to stop you from trying to go online and post your answers for other students. They want to stop you from searching the internet. They want to stop all that. Plus, they're making it a little easier for you because they've broken up the task verbs. That question that I showed you on Thursday is not actually any different than the question that we had back in May of last year as a practice question. The whole difference between that question and the question I showed you on Thursday is that they separated the task verbs. So it will be that long because that's just the length of a normal free response question. What is happening to my screen? I can't think that fast under pressure. Then don't think that fast. Take it one step at a time. Take one part, think it through, and do it. And like, if you need to set a timer on your phone to tell you when there's 5 minutes left so you don't see that timer, you can easily do that. They've said before that you can move the timer down so you don't see it on the screen. But I would have a backup timer. I would have something that's going to tell you, hey, you have 5 minutes left, you need to submit. Because playing games with that 5-minute timer, no.
No good. A lot of people who have had problems have been waiting. So people who wait to the last minute and a half have been having their uploads. Upload as soon as you can once that 5-minute window starts. The other thing to keep in mind is that your submission will keep uploading even if you've already moved on to question 2. So you absolutely do not want to refresh your page. Just have some patience, trust that it's uploading, but don't refresh your page. The timer is going to elapse, move to question 2, and then a new timer will come up on the screen.
So this is about null hypothesis again, but specifically looking at the chi-square. So when you're doing a chi-square, your null hypothesis is going to be what you have in your expected column. So the fruit fly question from 2012-ish, when you said that the fruit flies would be equally dispersed, since there were 60 flies, you would find 20 in each region of it, because that would be your null hypothesis that they were equally dispersed. I can't get rid of the question. There it is.
Where can you find the released questions? That's a great
AP Central.
Yes.
I can't. I lost it.
It's about 10 questions. Yeah, so the parts of the question, they correlate to points. So if you have, well, for example, the A to D is what the parts are, but there's going to be a certain amount of points within those. And your points are coming from what you answer through your action verb. So your describe, explain, predict, justify, that's where you get your points from. So in a lot of ways, how many quote unquote parts there are, it doesn't really matter. It's those action verbs. That's where you get your points, and that's what you want to address. We said this in the last session that we had together, but do your prep ahead of time. Meaning, if you plan to type your responses, then get a Word doc or a Google Doc or whatever you're using prepped in advance. Put your APID and your initials at the top. Write out A, B, C, D, so on. Get yourself ready so that on exam day, all you have to do is go into that document and write your responses. It'll give you a little bit of less anxiety. You can answer for question 1.
Someone wants to know about phylogenetic trees and cladograms. Those would be unit 7, so they should not be on your exam.
Yeah. Okay, so let's just say you are a speed demon and you are done for first question in 15 minutes. Good job. You still have to wait. So if you finish early and you have time left, a new timer is going to appear on the screen and count down for you again before FRQ2 will come on the screen, because this is a worldwide synchronized test. So you have to make sure they want you to wait to get to that next question until everyone gets to that question.
I also don't think that the submission box pops up until you have 5 minutes remaining anyway, so you can't submit early. So take your, if you're a speed demon, you're done in 15 minutes, look through your response, make sure that everything that you want to say is in there. Don't just try to sit down and do nothing.
You won't be bored on Monday. I think that we can guarantee.
You will not be. So sodium potassium pump is an example of active transport. So earlier we were talking about active transport, and active transport is going against the concentration gradient. So with the sodium potassium pump, we're pumping sodium out of the cell and we're pumping potassium back into the cell so that we can create a concentration gradient that would allow for a process that happens in the nervous system. And so the way that it works is literally that you attach a phosphate, which allows 3 phosphates to go out. The phosphate comes off, which then causes the protein to change shape, which brings the 2 potassiums back in.
Yeah, so the type of co-transport. And one of the things that it's a good example of is the fact that ions can't cross the semi-permeable membrane because they're charged. So we have to use a protein. Yeah, so the timer. So originally when the College Board talked about the timers, it told you that it was 25 minutes and 5. But when you actually log into the test, you're going to see a 30-minute timer because it's going to count down. But when it gets to 5 minutes, it will change to red, and that is your cue to submit. It will also flash on the screen to let you know that the 5-minute submission window is started. And finish your sentence, but then start to submit from that point forward.
So transcription factors, wanting to know, like, why are they significant? Transcription factors are going to aid in the RNA polymerase binding to the DNA to be able to transcribe. And so they can either be activators or they can be inhibitors. If they're activators, they're turning on the gene by making it easier for RNA polymerase to bind. If they're inhibitors, they're going to make it harder for it to bind. These would be found just hanging out in the nucleus, or they could be hanging out in the cytosol and they get transported into the nucleus. And it's usually due to a signal transduction pathway. It tends to be the response that we have activated this transcription factor, and now the factor will go bind to the DNA.
Yeah.
Sorry, I'm just trying to get through the questions.
There can be multiple transcription factors that have to come together in order for that to turn on. So sometimes it's not just the one. You might need like 4 or 5, and they fit together like a nice little puzzle so that RNA polymerase can come bind as well.
So let's kind of reassure you a little bit here. What if you don't know anything about the question? If you don't know anything about the question, I bet you know something, okay? Whether it is a word that's in the prompt that you're able to then expand off of, or whether you're just analyzing the data, okay? Especially if it says predict. Predict, you can give increase, decrease, stay the same, and you at least gave a guess, okay? Even if it's not right, you at least put something on the paper and you tried. And a lot of times, some of the questions I've seen in the past, predict, it's kind of wide open. If you had given any of those answers, you would have gotten credit for it. So just do your best, take a deep breath, and find something in the question that you do know something about to be able to apply. Do you have anything you want to add?
The key thing, something in the question. Don't like meander down a pathway and start defining a theory for them. You want to try and link it to the question if you can. What can go through the nucleus pore? Well, think about what needs to be in the nucleus and what has to leave. So mRNA transcripts absolutely can go through the pore. Small nucleotides, ribosomes can go through the pore.
A good example of how College Board can merge things together. A good example would be there was a question about muscles 3 years ago, in which you graphed allele frequency. So allele frequency was there. From the allele frequency, they then went to osmolarity, because if you had a higher concentration or more of the allele, you had a higher concentration in the cells. So they then got to osmolarity. From osmolarity, they then moved to evolution, talking about why some of them were more favorable than others. So they can kind of bring things together just based on kind of natural logical progression. Let's see what other questions we have. My button doesn't want to work. So someone, if there's math, do you have to show your work? Sometimes you do, sometimes you don't. It'll tell you if you need to show work. So it'll say calculate this, and then it says show your work if you need to show your work. I'm going to make the assumption that they probably don't need you to show your work on this because of the fact that it's potentially going to be typed. So it's kind of hard for you to show your work always with a typed thing. So you may or may not. And I think I heard from someone telling me that the chemistry test actually had no math on it. So you may not actually have math. It's hard for us to predict what's going to happen. I like to think that you're probably going to have a rate question in which it's going to give you a line graph and you've got to tell me the rate. And my students have heard this 20 million times. Rate is slope, guys. You can use. See the questions that we have.
So the exam goes from units 1 to 6. So it does not include evolution, which is unit 7, nor ecology, which is unit 8. However, evolution is a big idea. So it can be integrated into a lot of different topics, and they can really ask you anything as long as they give you enough information.
So the question was about types of mutations. That is totally a fair game thing because that can be a disruption that occurs. So I would definitely make sure you know the types of mutations because they may tell you that there was a frameshift mutation,
Yep.
assuming that you know what that is, and then ask you a question about it. Again,
be careful. Don't say mutations cause denaturation, because that's an assumption. And yeah, I understand. You want to go from DNA is different, and so the protein falls apart. But that's not really what always happens. And so remember, mutations can be neutral, negative, or at times positive. I mean, that's how evolution happens. DNA changes. And that's really what this means. It's a change. So some mutations are nonsense. Some have a greater effect than others. But try not to leap to those conclusions. Follow the breadcrumbs. So you change your DNA. You want to look, okay, does this change my transcript? Does that change my amino acid? Does that then change my folding? Okay, domino effect down the line. And when you look at your codon chart, so many of those amino acids are repeated, right? And so that's going to allow for the possibility of not having a change. Usually in the third letter of a codon, though. I can't do chi-square.
Yeah, I'm going to post something later that might help you with your chi-square. I'll post it to my story later for you guys. So specific examples about epigenetic inheritance, I'm kind of thinking about methylation. The big thing about the methyl groups is that when you add them to the DNA, it's going to condense the DNA, which makes it so that the RNA polymerase is unable to bind. And so if you inherit methylated DNA, then that DNA is already inactive, and so then you're not going to express that gene unless it gets unmethylated or demethylated.
Yeah.
So I think that we need to kind of calm this down, and maybe about 5 minutes we're gonna cut off. So like, you know, last minute questions that we have before we kind of log off. So I'm gonna say at 3:15 we're logging off, guys. So any last minute questions you have, throw them up there.
All right, absolutely, yes. If you have to use the codon chart, they will give it to you. You do not have to have that memorized by any stretch of the imagination.
This is a tricky question. So electron transport chain, yes, it is in the mitochondria. Yes, it's in chloroplast, but it's also in the plasma membrane of prokaryotes. Okay, and a lot of students don't know that.
Yeah, when you think about, so it makes sense though. Like think about when and where our chloroplast and mitochondria came from. And if you have the electron transport chain in the inner mitochondrial membrane, well, that was the original prokaryote membrane, right? So this makes sense. When you put your brain in that space, it makes sense as to why prokaryotes can do that too.
Okay, so any more questions that we have in here that we have not addressed? We've addressed, here's one we haven't addressed. Difference between water potential and osmolarity.
The greatest value of water potential is 0. That I can tell you.
It can be positive.
It depends on how it's applied.
Water potential can be positive. It's just the potential for water to move. And so water potential takes into account the solute potential and the pressure potential. And so when you get a shot, that's a positive pressure potential. And so if that's higher than the solute potential, it's going to force the material into the location. But the big difference between water potential and osmolarity is just looking at osmolarity is the concentrations of the solutes versus water potential is the potential for water to move. They are married and they relate to each other, but they are actually two different concepts completely. Okay. Any last minute questions y'all have? Because I got everything that's in the feed, like the question box.
I froze again.
I froze? That's not good. Thank you. We hope you do well on Monday. So I guess with my last 3 minutes, I'm going to give you, you know, my advice. Okay. Number 1, get on at 1:30. Well, my time is 2 o'clock. So 30 minutes before your exam, log on to the system. You're going to be answering some questions about yourself. It's going to kind of get you some information And it's also going to kind of see their writing style. So 30 minutes before, get it logged on. Even if it doesn't take you 30 minutes to answer the questions, if you get on 5 minutes before and it takes you 10 minutes to answer those questions, you're losing 5 minutes of your start time. So at the start time of the exam, it'll automatically move you into the questions or the exam. You'll get the first question. As you said before, there'll be 30 minutes that you have on your timer, 25 minutes for the question, 5 minutes to submit. Do your best. Read through the question. Read through all the components you need. If you don't know something, skip it. Okay, you can answer them out of sequence. You can do B, then D, then E, and then come back. Have a sheet of paper next to you so you could jot notes down, because it will be on a computer. You're allowed to print it, but it'll be on the computer so you can jot some notes down if need be. And of course, make quick notes. Increase this, decrease that, or this affects that, or this specific chemical does this. And that might help you so you don't have to keep reading the prompt over again, okay? And at the end of the day, you guys just do your best. Leave it all on the exam, okay? And when you get your score back, remember that you did your best, okay? And your best will always be enough, okay? If you do your best, there's nothing that any single person can say to you about your exam. You did your best. You gave it your hardest. You tried, okay? And that's all that anyone is asking you to do, okay? So, and as APsolute Recap just said, it's just a test. It's literally one day, one 45-minute time block in your day, and it does not define you. You've had an entire year of bio. Well, you've had a bunch of months of bio. And this will be a good support for you when you go to college. Even if you don't pass the exam, you still have all of that content support to make it a little bit easier when you do take the exam. And last thing I'm going to say is you guys are all going to be penguins on Monday. And so penguins are dressed for success. And so you guys are going to rock Monday because you are penguins. So thank you for tuning in. And I will continue to be posting answers to different people's questions on my feed. So if you have any additional questions, there's a question box on my story that you can pop your questions into, and I'll answer them the way I've been doing it basically since February. Have a good day. Bye, guys.